How carbon sequestration is modulated by mycorrhizal bacterial soil symbionts. 31% of our planet is covered by forests, but their distribution and abundance depend on a critical symbiotic association between mycorrhizal fungi and trees. Nevertheless, soil bacteria have been observed to have a crucial role in the establishment of that symbiotic association that has been overlooked. At the microscopic level, this association seems to regulate many aspects of the fungi tree physiology, including their capability to modulate, sustain or trigger growth and carbon sequestration. But how and what physiological variables determine the establishment of mycorrhizal bacterial symbiosis and which species are favored over other ones? At the lab, we have established protocols for addressing these questions through several techniques, including tissue cultures. Our experimental models comprise coniferous trees of economic and ecological importance such as sequoias and abbeys. The scope of this project is to identify the species of bacteria that promote mycorrhizal association with trees and most importantly which are the environmental cues that have an impact on the ability to accelerate the growth rate and carbon capture metabolism of abbeys plants. We would like to understand the metabolic regulation networks at the protein expression level and genetic regulation level by profiling the isolated symbionts complexes during their early stages of life by addressing their spatiotemporal successional distribution. Tailored designed microfluidics devices coupled with environmental sensors will help us to isolate, identify and process the bacterial mycorrhizal symbionts. The isolated complexes will be subject to DNA barcoding, rapid genetic expression profiling, and RNA capture using prepared magnetic nanobeads, to mention a few techniques. For the rapid identification and implementation of multiple experiments, we will use automated tools that we have previously developed at the lab that include robotic manipulation of samples, object recognition of tissues and cells, and the development of custom-made sensors and programs for the analysis of several physiological variables in living cultures. Want to know more about us? Visit us at www.glyxonbiolabs.com.